Welcome to the next unit, and in this unit, you will learn everything you need to know about cellular energetics.
Now, enzymes are made of proteins, and proteins are made of amino acids. Okay, and we have learned about the different types of amino acids. We get non-polar amino acids, which are hydrophobic. We have polar ones that are hydrophilic, meaning it will interact with water. We also have ones that have a positive charge and some that have a negative charge. Okay, these are all the different R groups that we find. So we start out with the sequence, and then we get the folding, and then the final 3D shape. Now, once we have that final shape, that shape determines the ultimate function of that enzyme. So structure determines function. When it folds into its 3D shape, it will have an area that we refer to as the active site. And this active site has different forms of chemistry. Okay, so some parts of that active site might be polar, some parts might have a charge, a positive charge or a negative charge. And that is why an enzyme is so specific to different substrates, because those different areas need to match up. Now, let's zoom in on the enzyme structure. So the enzyme has an active site region. So this is the active site region. This is where the substrate binds and catalysis occurs. Okay, so this is where we'll have the substrate that's binding. And here you can see, I'll color this in red. Okay, this is the substrate. Now, it is very specific to that enzyme. So let's say this enzyme has a polar, and it has a positive charge here and a negative charge here, positive charge, a positive charge. It would interact with the substrate's charges. So this substrate might have a negative charge there and a negative charge, and a positive charge, meaning that that active site is specifically binding to a very, very specific substrate. So the substrate shape and the chemical properties must be compatible with the active site. If it's not compatible with the active site, if the molecule is too big, it can't fit. So it needs to be the right size, and it needs to have the correct chemical properties to bind to that active site. Now, enzymes also have a second part within the active site. We have the binding sites, and then we have the catalytic site. Now, the catalytic site reduces the activation energy. So that's where the chemical reaction will actually take place. So here we can see in the induced fit model, this substrate can bind to the enzyme. Here it's binding to the active site of the enzyme. Now, once it binds, there would be a conformational change, meaning you can see that the enzyme itself is changing shape. So the shape of the enzyme adjusts to improve the substrate binding. Here we can see this is a red blood cell and how it has a conformational change when there is oxygen attached or no oxygen attached. The enzymes are 3D dynamic molecules. So this is why we learn about the induced fit model, because we understand that enzymes, their 3D structure can change to better allow the substrate to bind. And that change is a conformational change. So historically, we refer to the lock and key model, where we kind of assume that the active site and the substrate are already a perfect fit, and they have like this lock and key interaction. So, however, okay, we have learned that enzymes are flexible and they undergo a conformational change upon substrate binding. So as soon as the substrate binds, there would be a conformational change in the enzyme to better bind to the substrate. So the lock and key model is historical, and we now only learn about the induced fit, meaning we have that conformational change. If you like that lesson, don't forget to like and subscribe, and I'll see you for the next one.